Hi. Yeah. Yeah. Okay. So, okay. It's second stream. In first one, I did some mistakes. Hi everyone. I'm Aishman. Today we are going to start with the first chapter of biology IMAT, which is going to be cell for us. And the book we are going to use will be the NCERT from India, which is very popularly used for the NEET examination, which is also a very hard medical college entrance exam. And for now, I'm going to start because it's been night time and we should start now. I don't have an iPad today, so we are going to use the highlighters like this. Okay, so I'd say let's start working on our project, right? Okay, so we are going to start from the cell, the unit of life. The cell, it is a very very important chapter. We are going to learn about the differences between different type of cells, the similarities between different types of cell, the cells in the type of cell in microorganism, the type of cells in plants, the type of cells in animals. So according to me, the cell, the unit of life, it is a very fascinating and a very very beautiful chapter. Okay, so when you look around, and we are going like line by line. Okay, if there's any problem, I mean, just tell me. Although I know that there will be no students today, but okay, like everyone starts like this, right? Okay, so this thing is not so important. So okay, all organisms are composed of cells. We all know that every single organism is made of cells. Some are composed of a single cell and are called unicellular organisms. So organisms which are just made of a single cell, they are known as unicellular organism, and the organism with multiple cells are called multicellular organisms, right? Just wait a second. Mm, just wait. Yeah. So what is a cell? So you know what is a cell? The cell, it is a basic unit of life. Cell makes life come in existence, right? A cell is a unicellular organism which are capable of independent existence and performing essential life functions. So independent existence, what does it mean? Does it mean that the cell do not depends upon anything? No. The meaning of independent existence here is that a unicellular organism which do not depends upon the existence of any other living being, it is called a cell. Okay, the cell is like most basic thing which can survive on its own without the help of any other basic living thing, and performing the essential function of life. Obviously, if something is alive on its own, it must have the ability to perform the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Anything smaller than cell is not a living being. Like if we talk about virus, they are not considered a living being. Okay, why, we will talk about it later. And cell is the fundamental structure. Uh, Hence, cell is the fundamental structural and functional unit of life. Hence, cell is the structural because of it being the only the most basic structure which can have life, and functional because only it can carry all those essential function. It is the most structural and functional unit of life. And Antonio Leeuwenhoek. So there are three scientists about which I will tell you. First one being Robert Hooke, who for the first time saw dead cells on a wooden cork. And the second one being Robert Brown, who was the first scientist who told about the nucleus, which was obviously dead. And the third scientist is Antonio von Leeuwenhoek. He was the person who saw a living cell for the first time. Okay. The invention of microscope led to in many improvements leading to the microscope revealing all structural details of the cell. So, like Robert Hooke, he was able to look at the dead cells, by the help of a microscope. Obviously, because we can't see a cell by our naked eyes because they are so small in size. Okay, and by the help of microscopes, which were first discovered in the year 1950s. Okay, we are able to look at very structural details of the cells. Now we are going to talk about cell theory. So cell theory, like in early times, scientists were like trying to make theories around, because we all know scientists, researchers, they make different theories and different things, that these things are working, that how a black hole is attracting everything towards it, that how a sun is brightening so much, how it is burning so good. Okay, so here scientists try to explain cell and its lifestyle, its working. Okay, so Matthias Keldian, who was a German botanist, he examined large number of plant cell and observed that all plant cells were composed of different type of cells. All plants were composed of different types of cell, and that's right. Like in a plant, there are different cells on different part. Like if you will look at look in the leaves, there will be mesophyll cell, but mesophyll cell will not be present in stem, right? Which form tissues of the plants. Type of cells form different type of tissues, right? And at about the same time, Theodor Swan, who was a German zoologist, he studied different type of animal cells and reported that cell has very thin outer membrane, which was called plasma membrane. And he also concluded, based on his studies on plant cells, he also studied about plant cells, and he concluded that the presence of cell wall was a unique feature of plant cells. So, like I would tell you one thing, that when we are studying this, we are like, how should I explain this to you? So, guys, when you talk about IMAT, sorry, I don't have my iPad, so tolerate it. Okay, we talk about IMAT and we talk about NEET. So, like I am an Indian student, so obviously I'm going to talk about NEET. Okay, so IMAT and NEET, they are very different. And in which things are they different? So in IMAT, as much I have seen, they will not talk about the plant cells. Okay, they don't give a fuck about these cells. Okay, they don't give a fuck about plants. What they care about, they care about human physiology. Okay, they care about genetics, they care about the biomolecules, they care about biotechnology. Okay, they care about your mind, your ability to solve problems, not your ability to just put things in your fucking mind, and you don't even know how to use them. It's very different in IMET. Okay, in and on the other hand, in it, like you are just going to remember a lot of fucking things, like especially which I hate are like animal kingdom, plant kingdom. Oh my god, I hate those fucking chapters. So that's, I would say, a very very huge benefit of need. Sorry, a huge benefit of IMAP. Okay, so if we are going if we are talking about the scientists, believe me, they are not so necessary. You don't need to study about them. Okay. And now you'll say, okay, then what is very essential in the cell theory? In cell theory, the essential thing is that the cell wall is not a feature of animal cell, because we all know that cell wall is not a unique feature of plant cells as well. Okay. Cell wall is present in mm, other unicellular organisms. Okay. Like prokaryotes. Prokaryotes have cell wall. Like fungi. Fungi have cell wall, right? Now, they also can be based on a study that the presence of cell wall. Yeah, I just told you that. Okay. And on the basis in they propose cell theory. Okay, so what I will tell you about the cell theory, the most important things are, the first one being all living organisms are composed of cell and products of cell. Like a cell, it is also composed of itself, like a cell and all the products inside it, all the chemicals it is producing. Okay. And a new cell arises from pre-existing cell. Okay. Okay, I'll just take some water because it's first time we're teaching people on a live stream. The type of take some water because it's first time we're teaching people on a live stream. Okay, so an overview of cell. Um, so you have earlier observed cell in a onion peel or human cheek cell under the microscope. Not relevant. Let us recollect their structure. The onion cell, which is typically plant cell, onion is a plant cell, right? Onion has a plant cells has a distinct cell wall as its outer boundary, and just within it is the cell membrane. So, in case of plant cell, mm, there is a thick cell wall, and inside is a thin plasma membrane. Okay, but in case of human cell, there is no cell wall. There's only a single plasma membrane. The cells, the cells of the human chick have an outer membrane as a delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called nucleus. So, in not in prokaryotic, in eukaryotic cells, the inside inside the cell there's a structure which is called nucleus is present. Okay, the nucleus is present inside the cell. Let me remove it. I don't like. Okay. And the cells of human chicks have an outer membrane as it is. Inside nucleus, it contains the chromosomes. Okay, sorry. Inside cell, this nucleus contains chromosome, which in turn contain the genetic material. Okay, so nucleus it has genetic material inside it, which is stored in the form of chromatin and chromosomes, depending upon different stage of life of the cell. Cells that have membrane-bound nuclei, they are known as eukaryotic cell. Okay, and cells which lack the membrane-bound nucleus are called prokaryotic cell. In both prokaryotic and eukaryotic cell, a semi-fluid matrix called cytoplasm is present. So, when you go inside the plasma membrane, you found this surface, right? Is not this surface, no, it this space is filled with some materials, some solid materials, some bags as well, but majorly it is filled with a aqueous fluid which is called cytoplasm. Cytoplasm is a semi-fluid matrix, in which there are very important reactions occurring all the time. There are different transfer occurring from cell to cell, or from one part of cell to the another part of cell. Okay, various chemical reactions occur in this, keep the cell in living state. Okay. Besides the nucleus, the eukaryotic cells have other membrane-bound distinct structures which are called organelles, like the endoplasmic reticulum. In the nucleus, beside the nucleus, in case of eukaryotic cell, nucleus beside the nucleus, there are things like endoplasmic reticulum which are also present there. Okay, and also like the Golgi complex, which is very close to endoplasmic reticulum, the lysosome, which is produced by Golgi complex, the mitochondria, which is not dependent upon these three things, the microbodies, which stores food in them, vacuoles, which stores food in them. Okay, the prokaryotic cell lacks such membrane-bound organelles. So all of these, the endoplasmic reticulum, the Golgi complex, the lysosome, the mitochondria, the microbodies and vacuoles, they are membrane-bound organelles. But in case of prokaryotic cell, they do not have membrane-bound organelles. Then what the heck do they have? They have ribosome, which are also present in eukaryotic cell. Ribosomes, they are not membrane-bound organelles. They are like two big units like this. No, I mean one big and one small units like this, string together without being covered by any sort of membrane. Okay, ribosomes are non-membrane bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes found not only in the cytoplasm but also within two organelles. Ribosomes are found in cytoplasm in both prokaryotes and eukaryotes, but in case of eukaryotes, they are also found within two organelles, which are known as chloroplast and mitochondria on rough endoplasmic reticulum and on rough endoplasmic reticulum. So, does prokaryotic cells endoplasmic reticulum have ribosomes? The answer is no. Why? Because there is no endoplasmic reticulum in a prokaryotic cell. Prokaryotic cell do not have any endoplasmic reticulum, so they can't even have ribosomes there. Where are the ribosomes of a prokaryotic cells present? In the cytoplasm. Where are the ribosomes of a eukaryotic cells are present? They are present in on the endoplasmic reticulum. On which endoplasmic reticulum? Rough or smooth? They are present on the smooth endoplasmic reticulum. And where else are they present? They are also present on the mitochondria. They are also present on the chloroplast. Okay. Animal cells contain another non-membrane bound organelles which are called centrosome, which helps in cell division. So when you talk about cell division in animal cell, there are called centrosome, which are like pair of two cylinder-like bodies. These are the additional mm, membrane-bound organelles which are present in a, I'd say, in an animal cell. Wait a second. Mm, yeah. Okay. Cells differ greatly in size, shape, and activities. Uh, of course, every cell have different function. It has different size. It has different shape. For example, mycoplasma. It is the smallest cell. It is a prokaryotic cell, and its size is roughly near zero point 0.3 micrometer in length. Okay, my PDF is a bit lagging. And its size could vary from 3 to 5 micrometer. The largest isolated single cell is the oocyte cell, right? There's a size difference. One is a very small cell, the smallest cell, and whose size ranges from 3 to 5 micrometer, and the other one is the largest cell. So there's a very big size difference between two cells. And among multicellular organisms, human RBC are about 7 micrometer in diameter. Nerve cell, they are some of the longer cell. So like look, there are four type of cell which has different properties. One is smallest, one is largest, one is a normal size cell, and one is a longest cell. That's different sort of cells, and all of them have different function. Like mycoplasma, it is a unicellular organism. Like oocyte cell, it is going to be fertilized. Like RBC, its function is to transport oxygen and nutrients to the human in the human body. Nerve cell, its function is to transfer signals from one part to the another part of the human body, right? Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, and thread-like, and they can even have irregular shape. The shape of the cell may vary with the function they perform. Um, this I can tell you. Like if you look at a nerve cell, I mean, what does this statement says? This statement says that the shape of a cell, the particular shape a cell has, it can vary according to its function. Like when you look at a nerve cell, its shape is like a branch shape. Why? Because it has to transfer signals from one part to another part, from multiple source to single source, from single source to multiple source, from multiple sources to multiple sources, right? And like you can look at RBC because it has to travel from sometime very narrow spaces. So its shape is like a disc shape with also a biconcave structure, right? So that it can easily pass through the narrow pathways. Um, yeah. Now we will study about prokaryotic cell. Prokaryotic cell, they are represented by bacteria, Beugen algae, and mycoplasma and PPL. I'm going to stop the stream, like not stop the stream. I'll just wanna mute it for once. Okay, hey, I'm back. I'm back. Yeah. Mm, yeah. So now we are going to study about prokaryotic cell. The prokaryotic cell, they are also represented by bacteria, Beugen algae, mycoplasma, and PPLO. Uh, by bacteria, we mean different sort of bacteria, blue-green algae. So, blue-green algae, they are sort of bacteria which stay in group, and they are called as brewing algae. Mycoplasma, we just talk about it, very small organism, and PPLO, pleuropneumonia like organism. Okay, they are generally smaller and they have multiple they multiply more rapidly than eukaryotic cell because they have a very small size. They don't have too much functioning to be done to reproduce the cell, to divide the cell, so they divide much rapidly than a eukaryotic cell. They may vary greatly in shape and size. The four basic shape of prokaryotic cell are bacillus, they are coccus, they are vibrio, and spiralium. Bacillus is like rod-like, coccus is like spherical, vibrio is like comma-shaped, and spiralium is like spiral-shaped. Um, the thing which I'm going to repeat as I repeated it earlier, every single information is not required for IMAT. You have to attempt the previous year questions, then you will come to know that okay, what kind of question the organization is asking me to enter in their college. Okay, what kind of question are asked in this examination to qualify myself through it. The organization of prokaryotic cell is fundamentally similar, even though even though prokaryotic exhibit wide variety of shape and function. So, the organization of prokaryotic cell is fundamentally similar. Okay, a prokaryotic cell is fundamentally similar, but still they have a variety of shape and functions. All prokaryotes have cell wall. All the prokaryotic cells have cell wall. So it also means that plant cell is not a specific function. Sorry, plant cell do not have. Sorry, cell wall is not a specific function of plant cells. Okay, the semi-fluid matrix filling the cell is cytoplasm, which are also present in eukaryotic cells. There is no well-defined nucleus. Prokaryotic cells do not have a well-defined nucleus. The genetic material is basically naked. Their DNA, it is not like surrounded by nucleus walls. It is basically naked, but still it is wrapped around non-histone proteins so that it do not acquire too much of the space. The genetic material is basically naked. It is not enveloped by a nuclear membrane. In addition to the genomic DNA, there is a single. Okay, so the genomic DNA is a single chromosomic circular DNA, and in addition, which means that genomic DNA is also present, but except that there's also a small circular DNA present, which is called as plasmid. The plasmid, it gives a different sort of phenotypic characters to the bacteria, being resistant to the being resistant to the antibiotics, and they also help in bacteria transfer. They also help in monitoring the bacterial transformation. Okay, the nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cell. So the nuclear membrane, it is only found in eukaryotes. It is not in prokaryotes. The organelles, what sort of organelles? The membrane-bound organelles. They are also not present in the prokaryotic cell. They are only present in the eukaryotic cell. A specialized differentiated form of cell membrane called mesosome is the characteristics of prokaryotes. They are essentially infoldings of the cell membrane. So in case of prokaryotes, there are infolding like this. Like this. They are infolding. Infolding means the folding of cell membrane inside the cell. Okay, like this. This is inside the cell. Okay, these are called as mesosome. And mesosome, they carry essential functions for the prokaryotic cell, like respiration, like um, transfer of different materials inside and outside the cell. Okay, Now we are going to study about cell envelope and its modifications. So in case of prokaryotic cell, there are three layers: first one, second, and third. The third, which is the inside one, the most inside layer is cell membrane, or I say plasma membrane. The second one being cell wall, and the fourth and the third one is the most outermost is the glycocalyx. Okay, so both of three together they makes the cell envelope of a prokaryotic cell. Most prokaryotic cell, particularly the bacterial cell, have a chemically complex cell envelope. The cell envelope consists of tightly bound three-layer structure. The first one being the outermost glycocalyx, which is followed by cell wall and the plasma membrane. Although each layer of the envelope perform distinct function, all of these layers have different function. Like cell membrane, or I say plasma membrane, its function is being semi-permeable and deciding which things does a cell need and which it don't. Cell walls just keeping it compact, and glycocalyx protection. But all these three layers collectively, all these three like glycocalyx, plasma membrane, and cell wall, all these three collectively, what does they do? They form a single protective unit. Bacteria, it can be classified into two groups on the basis of difference in cell envelope. Okay, and the manner in which they respond to staining procedure developed by Gramwitz. So on two bases, first one being the difference in the cell envelope, and second one being the manner they in which they respond to the gram staining. They can be differentiated into two parts in the each case. So in case of gram staining, they can be described as gram positive and they can be described as gram negative. So when we apply stain on them, and if they takes the stain, then they are called gram positive, and if they do not take the stain, and when they are washed they are present without stain, then they are called gram negative. Okay, and on the basis of cell envelope, the first one being if glycocalyx differs in composition and thickness among the different bacteria. Okay, it can be loose sheath. In that case, it will be called a slime layer, while in other case it can be thick and tough, and then it will be called a capsule. Okay, the cell wall, it determines the shape of the cell, and it provides a strong structural support to prevent the bacteria from bursting or collapsing. So, as I just told you, that the function of cell wall is to just keeping the structure of the cell together. Okay, it do not let it collapse, and it do not let it burst. And the function of plasma membrane being selectively permeable to decide what will go inside the cell and what will stay outside of the cell. And now we are going to study about mesosome. As I told you, mesosome are very important extension of plasma membrane into the cell, means enfolding of the plasma membrane into the cell, right? These extension, they are in the form of vesicle, tubules, and lamellae. There are three sort of extension of plasma membrane, or three sort of mesosomes. Vesicles, if I can see, there are any sort of vesicles. Like this one is almost going to be a vesicle, right? And tubules like this one, it is like a very long tubule inside it, right? And one being like the lamellae, they help in cell wall formation. Like just look at all those functions, like cell wall formation, like DNA replication, distribution to daughter cell, and they also help in respiration secretion processes to increase the surface area of the plasma membrane enzymatic content. So mesosome is one of the most important structure in a prokaryotic cell. Okay, and in some prokaryotes like cyanobacteria, there are other membranous extension into the cytoplasm, which are called chromatophores, which contain pigments. Okay, in prokaryotic cell, like cyanobacteria, there are other membranous extension Like if you look at mesosome, it is a membranous extension. So there are other sort of membranous extension in some sort of cell, like chromatophores, which have pigments in them. And what does the pigment do? It gives colors to the cell. Bacterial cells may be motile or non-motile, and bacterial cell, they can maybe move freely or they can't move freely. And if they are motile, if they have ability to move, they will have a thin filamentous extension from their cell wall, which is called as flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagella is composed of three parts: posterior filament, hook, and basal body. The filament is the longest part, okay, the long tail, and it extends from the cell surface. It extends from the cell surface. It means it is extended from the glycocalyx, because obviously the cell surface is made up by the glycocalyx. Beside flagella, pili and fimbriae are also some surface structures of the bacteria. Although they do not play any role in motility, the pili are elongated tubular structure. So the pili, they are made of special proteins, and they usually help in connecting one bacterial to another bacteria, and they also help in a process called conjugation, in which there's a special plasmid which is called F1 plasmid, which is transferred from one cell to another cell. And the fimbriae, they are small bristle-like fibers which are sprouting out of the cell. In some bacteria, they are known to attach the bacteria to rocks in stream, and they also in rocks and streams, and they also to the host cell. Fimbriae, what does they do? They help a bacteria cell to adjust to rocks to host cells, right? Now we are going to study about the ribosome and inclusion body, which is going to be the last topic of this lecture. I hope that I will be able to do a very big lecture the next time, but this time we are going to end it just at a near mark of 40 minutes. So, ribosome and inclusion bodies. In prokaryotes, ribosome, they are associated with the plasma membrane of the cell. So, as we know that in prokaryotic cell there are no extra structures present on which ribosome can be found. So ribosome will be found only in cytoplasm, floating in cytoplasm. The second one being associated with the plasma membrane of the cell. Their size is about 15 to 20 nanometer. They are made of two subunits, first one being 50s and the second one being 30s. And they together they make 70s subunits. Okay, these are the only type of ribosome found in the prokaryotes. So the 50th subunits, it is generally larger, and the 30th subunit is smaller. Ribosome, they are the site of protein synthesis. Ribosome does protein synthesis, and several ribosome may attach to single mRNA. So, if you don't know, a mRNA thread is being used in the protein synthesis, and what does they do? Like several ribosomes attach to a single mRNA to make a chain, which is called polysome or polyribosome. The ribosomes of a polysome translate the mRNA into protein. The different codons of the mRNA, they are translated into the proteins. The ribosome of a polysome, what does it do? They translate the mRNA into the proteins. The inclusion bodies. Now we are going to study about inclusion bodies. They reserve material in prokaryotic cell. They are stored in the cytoplasm in the form of inclusion bodies. They are not bound by any sort of membrane system, and they lie freely in cytoplasm. So, in plant cells, if you know, the excess material is stored in the vacuoles, right? In similar case, in prokaryotes, they are stored in the form of inclusion bodies, which are not bound by any sort of membrane. They just lie and float freely in the cytoplasm. There are different sort of inclusion bodies, like phosphate granules, like cyanophysin granules, like glycogen granules. Gas vacuoles, they are also found in blue, green, purple, and green photosynthetic bacteria. Gas vacuoles, they are also some sort of inclusion bodies. Okay, they are also some excess materials stored inside the cell, which are found in like blue, green, purple, green photosynthetic bacteria. Okay, so I think for today it is enough, guys, because like my nose is running a lot of water through it, so I can't teach you guys so well. Although I will try to teach you guys much more effectively tomorrow. For today, the lecture is over here. Tomorrow we are going to study about these diagrams because I think we skipped the this diagram, right? But I don't know. There's too much information in it because it is not so much clear. Tomorrow we are going to study about eukaryotic cell. These diagrams as well. Then we will be studying about cell membrane. Then we will study about cell wall. Yeah, this topic endomembrane system. It is a very good topic. We will study it tomorrow. Okay, so tomorrow we are going to focus on these things. Until then, bye bye. Take care, everyone. Okay, and have a good day. Study well. Your exam is coming soon, so please be prepared for it. And bye bye, guys.